Hi learners, welcome to yet another presentation of ELISA in which we will discuss about the advantages and disadvantages of ELISA. So it is the concluding part of our ELISA series. Four major advantages of ELISA that it is versatile, simple, sensitive and quantifiable. So the simple procedure is there for ELISA and it's easy to do. It involves two antibodies. Test result is an accurate diagnosis, high specificity and sensitivity. Fine diagnosis in complicated cases as antigen is not required to get purified to detect. As direct indirect analysis method are involved, it is highly responsive. Not a complicated method as it does not involve the presence of radioactive materials and large amount of organic solvent. Equipment are inexpensive and widely used method compared to others. But disadvantages are also there, like it's a labor intensive, finest technique and expensive to prepare the antibody. To obtain a specific antibody, culture cell media are required. Plasma constituents may affect the activity of the enzyme in the sample. More chances of false positive or negative result. Instability of antibody in the sample. Storage and transport in cold media is required. Kits are commercial available, but they are not cheap. Test is specific to a particular type of antigen and can't detect other antigen in the sample. There are limitations also that result may not be absolute, availability of antibody not possible all the time, false positivity due to long-standing primary antibody or non-specific binding of antibody or antigen in the given sample, and there's a chance of false negativity. Advantages and limitations of ELISA. In advantages, where we can run in the batches and it is suitable for the large number of samples. Quantity assurance, quality control is easier. Evaluation at national and regional lab only, and it will identify the seroconverters earlier. Whereas in limitation, it requires a skilled person for the interpretation. Testing time is more than 2 hours, and specialized equipment setup is required. Application of ELISA. So ELISA is applied in many diagnostic tests, like it is used to detect and measure the presence of antibody in the blood, autoantibodies, or antibody against the infectious disease in hepatitis A, B, C, HIV, etc. So it will detect and estimate the level of the tumor. It will detect and estimate the level of hormone like FSH, prolactin, testosterone. It will help in tracking the disease outbreak, for example, in cholera, HIV, and influenza. It will also detect the past exposure, whether the patient is having any past exposure of HIV, hepatitis. It help in the screening of the donor bloods against the HIV, HCV, hepatitis B, malaria, and syphilis. It will detect drug abuse in the patients. Thank you everyone. Like, share, and subscribe the video.